This video is an introduction to proteins, the most versatile biological molecules with the widest range of functions. So what makes them so special? Let's dive into the fascinating world of proteins. First things first, proteins are biological polymers. Their monomers are amino acids, which are joined together by peptide bonds formed through condensation reactions. That is, reactions that release a water molecule. Now there are two main types of proteins, fibrous and globular. Fibrous proteins like collagen or keratin are long and stringy. They're super tough and play structural roles in our bodies. Collagen helps remove dead skin cells and act as the scaffold of skin, providing strength and elasticity to it. Keratin, present in epithelial tissues, form the structure of hair, nails and skin. On the other hand, globular proteins like enzymes, antibodies and hemoglobin have a specific three-dimensional structure important for their versatile functions. From speeding up chemical reactions, providing specific immunity, onto transporting oxygen. But how do proteins get their unique shapes? This brings us to the four levels of protein structure. First is the primary structure, which is the sequence of amino acids coded for by the codon sequence determined by the alternative splicing mechanism. Next comes the secondary structure, which consists of coils of alpha helices and beta-pleated sheets that are produced when beta strands are folded next to each other. The third level, tertiary structure, is the result of interactions between amino acid residues brought close to each other by the formation of the coils and folds of the secondary structure. These interactions include hydrogen bonding, hydrophobic interactions, van der Waals forces, as well as covalent adionic bonds. The sum of these produce the full 3D shape of the protein. Finally, quaternary structure comes into play when multiple protein chains or subunits join together. A good indicator of the importance of proteins to living organisms is that the genes in the exon regions of every organism's DNA code the specific sequence of the amino acids of each and every protein our cells make. They tell the cell which amino acids to use and in what order. And the process of alternative splicing mentioned at the beginning of the video enables multiple different proteins to be produced from a single gene. This process and protein synthesis, as well as the functions of different categories of proteins, are covered in other videos. The diversity and versatility of proteins is the consequence of the different motifs and domains present within their structure. Motifs are small, recurring structures within the protein, an example being the beta strands mentioned previously. While domains are larger, stable, functional units that fold independently of the rest of the protein and are responsible for specific functions performed by the protein. Many of these functions are dependent on domain-domain interactions. Domains within a protein or between different proteins can interact with each other. These interactions are crucial for many biological processes like signal transduction and cellular communication. So, there you have it. Proteins are incredible molecules with a huge variety of structures and functions. From the amino acids that form their backbone to the complex interactions that determine their roles, proteins truly are the workhorses of the cell. Thanks for tuning in. If you like this video, hit the like button and subscribe to this channel. Also, hit the notifications bell to be alerted to all new videos published to this channel. Keep exploring, keep questioning and stay curious. See you next time.